Hello! I am Sophia, and today I am going to present the cell cycle. Here is a cell in a very particular stage of cell division, which is mitosis. You can already see how the cell is dividing. It has this nucleus and all its organelles right there straight, and it's right ending to divide. It's right, all its nucleus and all its chromatid is there. So it's a point where it's fairly late. Its mitosis has almost already finished. So this is actually not really in mitosis. This is kind of like at the very, very end of mitosis, where you've got the whole thing down. All of the nucleus. This is actually in like early cytokinesis. This is in early cytokinesis, late mitosis. Okay, so here we have the control of mitosis. But before we go to that stage, we are going to go back to Campbell biology and go through this important thing. Here is the very important resume. Why does cell division function like that? It's for asexual reproduction, for growth and development, and for tissue renewal. And here you see eukaryotic chromosomes, that the chromosomes are visible within the nucleus of the cell, and the thinner red thread, the cytoskeleton. It's preparing to divide. It's almost at that point of division. Oops. Ooh, nothing happened. Okay. So, here we have that. Very important topic. A little bit larger than it should be. Yes. Okay, so here we have a highly condensed and duplicated chromosome, which is through this so-called SEM, or the scanning electron, so-called the scanning electron microscope. And here you have the sister chromatids and the centromeres. There's one centromere on each sister chromatid. So here we have this very important topic of chromosome duplication during cell division, because during the mitotic phase there is an important stage here. That's how chromosomes duplicate. So one of the multiple chromosomes in a eukaryotic cell is represented here. It hasn't duplicated, and then it duplicates, including DNA replication and interphase, and it condenses. And that occurs until metaphase of mitosis. We'll talk about the phases of mitosis later in this video. And then once duplicated, that chromosome is consisted of two sister chromatids, and they're connected by sister chromatid cohesion, because they're splitting apart during anaphase. So the molecular and mechanical processes during anaphase, they separate the sister chromatids into two chromosomes, and they distribute them to their two daughter cells. Okay, so here we have the cell cycle, and in a dividing cell, this mitotic phase alternates with interphase, and the mitotic phase is constituted of mitosis and cytokinesis. Okay, let's talk about the control of mitosis. So here we have several different cyclins. So cyclin E is active between G1 and S. Cyclin D is less active, but is more active, or most active, during this period in the middle of the S phase. And the cyclin A is more active in the G2 phase, and the cyclin B is more active in early mitosis. So first we have this G1 checkpoint, which is the restriction checkpoint. So, how does it get controlled in this case? Why doesn't a cell divide at the wrong time? So a cell doesn't divide at the wrong time because there was this very important checkpoint. So, if in the absence of a go-ahead checkpoint, it goes to another phase not depicted here, which is the G0 phase. And if the cell receives that go-ahead signal, it continues with the synthesis phase and the G2 phase. And then there's this G2 checkpoint, where it checks if the cell is large enough and if the DNA is damaged or not damaged. And then what occurs after that, we have that metaphase checkpoint. So a cell in mitosis receives a stop signal when any of its chromosomes are not attached to spindle fibers, and that's in prometaphase. When all chromosomes are attached to spindle fibers from both poles, a go-ahead signal allows the cell to proceed into anaphase. Wow, that works. Here is a summary of mitosis. So here we have this chromosome A, chromosome B, and then we replicate our DNA. And then what we get is this cell and this other cell. And then they produce two diploid cells that are identical to the parent, which is also diploid. Okay, so here we have the cell cycle in animal cells. First, in interphase, we are going to talk about, so that at gap 1, G1, the cell grows. If it goes into the resting phase, G0, it leaves the cycle. In the DNA synthesis phase, or S phase, it duplicates its DNA and the centrosome duplicates. In gap 2, G2, the cell grows some more. Also, all of its organelles are duplicated. In prophase, the chromatin condenses into chromosomes and the nucleolus disappears. In prometaphase, the nuclear membrane breaks down. And the kinetochore microtubules invade the nuclear space and attach to kinetochores. And the polar microtubules and microtubules push against each other. They move the centrosomes apart. In metaphase, chromosomes line up along an imaginary plate, which is called the metaphase plate. In anaphase, the chromosomes break its centromeres and sister chromatids move to opposite ends of the cell. In telophase, the nuclear membrane reforms, the nucleoli reappear, and the chromosomes unwind into chromatin. In cytokinesis, myosin II and actin filament ring, they contract to cleave the cell into two. Now we have two diploid daughter cells. Now we have the plant cell cycle, and it's so unclear that I'm just going to show you the real figure here. First, we have interphase gap 1. The daughter cell grows and expands. The organelles like chloroplasts and mitochondria that aren't shown divide through interphase. The S phase is where the cell replicates its DNA. In gap 2, the nucleus containing loose chromatin in a nucleolus is embedded somewhere inside the cell's large central vacuole, and the transvacuolar cytoplasmic strands containing actin filaments radiate from the nucleus, cutting through the vacuole to connect the nuclear cytosol to the cortical cytosol. Mitosis. There's a pre-prophase, but you don't really need to worry because in this book it isn't depicted like the mitotic phases are different. But what I think the mitotic phases are prophase, prometaphase, metaphase, anaphase, and telophase. So we're always going to have prophase and metaphase and anaphase and telophase. So, but sometimes we have prometaphase, sometimes we have pre-prophase. This is the first place where I've heard of this. As the cell approaches prophase of mitosis, the cytoplasmic strands shorten, pulling the nucleus to the middle of the cell, forming a disk of cytoplasm across the division plane called the phragmosome. It isn't really important to remember the word phragmosome so much. Most of the cell's Golgi stacks move into the phragmosome. Along the edge of the phragmosome, actin filaments and microtubules collect to form the pre-prophase band. Inside the nucleus, chromatin condenses into chromosomes and the nucleolus disappears. The nuclear envelope generates the mitotic spindle, which is organized into two poles by the pre-prophase band. Okay, so first we have prometaphase, and I'm going to get a little bit smaller in this case. Okay. At prometaphase, the chromosomes finish condensing and the nuclear envelope quickly breaks down, followed by the pre-prophase band and its actin, leaving behind an actin-depleted zone Kinetochores form at the centromeres of the chromosomes, allowing microtubules to attach. Metaphase. After some of the spindle microtubules attach to all of the chromosomes, the chromosomes are aligned along an imaginary plane called the metaphase plate. About a fifth of the Golgi stacks in the cell migrate into the mitotic spindle area and start to secrete vesicles of cell plate materials, and another fifth gather where the preprophase belt used to be. The remaining stacks return the rest of the cortical cytosol. So what is occurring here is that anaphase is a very short phase that sometimes only lasts a few minutes. Okay, early anaphase. The cohesin proteins that hold the two sister chromatids of each chromosome are broken. The kinetochores on each sister chromatid pull them along the spindle microtubules, carrying the sister chromatids to opposite ends of the cell. Meanwhile, a structure made out of microtubules and actin filaments formed between them called the phragmoplast. Golgi stacks line up at the edge of the phragmoplast, releasing vesicles full of cell plate building materials called cytoglucans. Late anaphase. Guided by the microfilaments of the phragmoplast, the Golgi-derived vesicles align at the metaphase plate, it should be plate with a T, and fuse into a flimsy cell plate. Callose begins to be synthesized within the cell plate. The phragmoplast expands outward towards the cell wall. Mitosis and cytokinesis. Telophase and cell plate expansion. As the chromosomes unravel and nuclear membranes reform at opposite ends of the cell, the phragmoplast continues to move outward as the cell plate grows. Some phragmoplast microtubules reorient, pointing towards where the preprophase belt used to be. Callus fills the cell plate and the Golgi stacks add to the cell plate at the edges. Cytokinesis, cell plate maturation. When the cell plate membrane comes into contact with the cell membrane, the two membranes fuse and cellulose-synthesizing enzymes found in the cell membrane float into the fluid mosaic cell plate membrane. We've been talking about the fluid mosaic model in a video called Cellular Membranes, just in Chapter 7 of Campbell Biology. Making cellulose that replaces the callose in the cell plate. The Golgi stacks make the pectin that also goes in the cell wall. The new cell wall divides the parent cell into two daughter cells. Now it's done. It's the whole cycle. Imagine. That isn't so long a cycle, but it seems difficult. Oops, like that. And then here we have the whole thing. We see this is prior to mitosis, early prophase, late prophase, the doubled chromosomes in the centromeres, the early anaphase, the late anaphase, and here we have telophase and the completion of mitosis. They kind of ignore cytokinesis. Here we have it. There are three types of cell division. The first is binary fission. And first, I'm going to introduce binary fission. It's in a figure of Campbell Biology. So just as this is the whole cycle of mitosis. And we've already talked about cytokinesis. So, chromosome replication begins at the origin. Soon after, one copy of the origin moves rapidly towards the other end of the cell by a mechanism involving an actin-like protein. Replication continues. One copy of the origin is now at each end of the cell. Meanwhile, the cell elongates. Replication finishes. The plasma membrane is pinched inward by a tubulin-like protein, and the new cell wall is deposited. Two daughter cells result. Ooh, yay! We have the whole thing. Okay. Flip the page. Okay, so we have the whole thing. That's the binary fission. So this is binary fission, DNA replication, chromosome segregation, and cytokinesis. And then we have mitosis that we've just talked about before. Diploid cell, then S phase, then these are mitosis. And then we have meiosis that we're going to talk about in the next video. Okay, so here we have cancer. We have a small polyp that is soon going to become the large polyp, and the large polyp that is going to become this cancerous source. This is the source of cancer. Okay, quiz. Test your understanding. Okay, identify which cells marked as which letter are dividing. Identify all the depicted phases. Okay, I'm going to give you a little bit of time, and if you need more, then pause. Okay, now Don and I am going to go to the next slide that actually contains the answers. The answer is that the cell marked with the letter C are dividing. So this cell is dividing, this cell that isn't marked is dividing, and this cell is dividing. And the phases depicted are metaphase. We have this one's metaphase. And anaphase. This is anaphase. This is anaphase. This is a textbook from the 1900s, so it's quite old. Okay, so here we have some micrographs. Oops, don't look at this. Please don't look at the answer. I should have edited this so that it comes ahead. Okay, so now it isn't here anymore. Yeah, so please don't look at the answer. Just forget about the answer. So identify the phase depicted in each image and number them 1 to 5 and order them in chronological order. What you do is that this phase is number 1, this phase is number 2, this phase is number 3, this phase is number 4, and this phase is number 5. And you should declare them by which phase comes first, which phase comes last. This image does not contain cytokinesis. Okay. I'm going to go over a little bit of time and then, again, go to the next slide. Okay, I'm going to the next slide. So the answer is 2, 5, 4, 1, 3. Let me discuss. So first 2, this is prophase. And then 5 is prometaphase. 4 is so first we have this 2, which is prophase, 5, which is prometaphase, and then we have the number 4, which is actually, I'm going to check, because I might have made an error here. So this 4, we have the telophase. Apparently we have the prometaphase, so what comes next? So the order, you've already know this, so this, apparently, let me just check because I might have made an error here. That, yeah, this is prophase, and this is just like, I can't really order this one. What's wrong? Okay, so I'm just going to go back and pause this. Okay, so I realized the confusion was because I misnumbered things. So this is number 1, and this is number 2. And this is number 3, and this is number 4. So apparently it wasn't an issue in the answer. So actually, so number 2, this is prophase, and this is prometaphase. And then after that, this is, yeah, so then, yeah, this might be telophase, actually. So this is telophase, and this is prometaphase. This is the splitting of the chromatids. So what I probably did in an error, actually, was that confused it. So number 3 isn't telophase. So number 3 is actually metaphase. So it shouldn't be 2. So really what it was is a misordering. So this should have been 3, and this should have been 4 instead. So 3 and 4. So 3 is metaphase, 4 is telophase. So now a little bit of the correct order is this is prophase, this is prometaphase, this is metaphase, this is anaphase, and this is telophase. Okay, so these are the sources of the images, and then this is the second piece of the sources. Thank you. Bye.